good afternoon, everyone. Okay, I'm back with another lesson. Okay, so I've been not free the last few weeks, and yesterday I'm supposed to have a lesson actually on every Saturday, but I've already postponed it to today because I know that all of you are all very getting prepared for your exam, and of course, very soon you're going to have your Ujian Amali Sains. Okay, and I hope that my video will be able to help you answer some questions confidently. Okay, so today I'm going to discuss about your practical exam. Okay, good afternoon. I see a few students here. Okay, Shell Yu, Red Block, and Ria. Okay, good evening, good afternoon. Okay, thanks for joining the class. And later, after your lesson is over, you can actually share the link with your friends who have missed the lesson live. Okay, Matthew. Hi. Okay, good afternoon. Yes. Okay, so today I'm giving you a very general idea of what kind of questions or types of questions that you can be asked during this Ujian Amali Science. Actually, they can ask any experiment, okay? But bear in mind, the experiment that they can ask is only the experiment that can be done within 40 minutes. Okay, it cannot be an experiment that takes a lot of time. So by that, you can actually eliminate some of the experiments that you know is not possible to come out. Okay, all right. And then I see Jia Ying. Okay, good afternoon. All right, so I'm going to share screen now. All right, so I'm going to give you a few types of questions. Actually, there are only 11 types of questions. Okay, whatever experiment they give you, they can only ask these 11 types of questions. And what I want to do is actually to guide you. Okay, so if you're confident enough already, it doesn't matter what experiment comes out. All right, if you know how to answer this type of question, you know how to answer it, you can actually get it right. Okay, of course, you need to know your knowledge as well, your theory. Okay, then only you can answer correctly lah. All right, okay, so I'm going to start straight away. Yeah. All right, so today's lesson will not last more than 1 hour. In fact, it's going to be less than 1 hour. All right, I'll try to make it brief as possible, but I don't want to miss out anything. So I'm going to guide you through all the 11 types of questions. Of course, in any experiment, all the 11 types will not come out because you only have 15 marks. Okay, 15 marks, that means there will be some construct or questions that they will not ask. So we do not know what, okay? Unless, of course, you're in the Badan Penggubal, you're the one setting the question, right? We do not know that also. Okay, so let's go on. Now, just to give you an overview, because you have not done your Ujian Amali Science before, but you have done your exam in schools. Okay, just to recap, yeah. So 45 minutes only, and every question, they have a sub question. Okay, but actually there's only one question. The experiment is only one. There are sub questions. So sub questions can carry one up to 4 marks. 1 mark, 2, 3, or 4 marks. Maximum is 4 marks. Okay, then the planning, right? You're required to, actually you're not required to do experiments straight away. So the first 5 minutes is for you to check your apparatus, to check the materials that are provided to you, make sure that they work well, the apparatus is not faulty, all right? And you, if there's any problem with it, you test it within the 5 minutes, and you can actually get a replacement. Please ask for replacement, okay? So use your 5 minutes well to check your equipment. All right, okay. Now next, your answering time is only 40 minutes. Okay, so the first 5 minutes, you can't cheat by taking another extra 5 minutes because you're not allowed to start the experiment. So use that time to just check your apparatus, make sure they work well. Okay, so my slides are actually in doing bahasa. So if some of you are actually having taking your SPM in BM, doesn't matter. All my slides here, we have in English and BM. Okay, but I explained in English. 2 weeks ago, actually, I've already explained the Ujian Amali Sains, but in BM. All right, so there's also on my channel. So if you want to see the BM version, just find it on my channel. All right, okay. Now, and then I just stress here that you're not allowed to do any experiment. Do not start within the first 5 minutes. Okay, all right. So, yes, someone said something, red block. Okay, DLP class, yeah, all right, good. Okay, DLP class, then I'm speaking mostly in English. So for those who are non-DLP, you will see that 2 weeks ago, I think I've already done a video, same video, the same what you call points lah, yeah, but it might look slightly different, but mostly I speak in DM for that. Okay, now, what are the 11 types of questions? We call it the 11 constructs. Okay, 11 constructs, so I'll go through very quickly first, and then I'm going to zoom in each one of them. Okay, and then I give you example, and then hopefully you can catch or what is needed and how to write the answer. Okay, so the 11 constructs here is, number one is you may be asked to write the procedure. That means instructions are not given to you. They give you the apparatus, they give you the materials, the specimen, whatever. You need to know what to do with them lah, right? So you need to write the procedure. Then you also need to, they're testing these skills. These are the skills. These are called the science, process science skills, all right? All scientists should have this kind of skills. So you are also a science student, right? You should have these skills. The next thing you need to do is how to construct a table. And constructing a table, you must have your manipulated variable, your responding variable. Okay, must exist in the table. Okay, making observation. How do you make an observation? What are you going to write when you make observation? Okay, next. After making observation is usually followed by the question called making inference. So after observation is inference. Okay, inference basically just means why do you get this observation? You're supposed to give an explanation. Why do you get this? Okay, your observation is you state lah. You state what you see, what you observe. All right. The next question, observation, inference is you explain why you get this result. Okay, very simple. Okay, after the give example. Right, listing variables, you need to know how to list out all the three variables: MV, manipulated variable, responding variable, and constant variable, fixed variable. Next, making hypothesis. Okay, you need to know how to write a correct hypothesis or an acceptable hypothesis to for your experiment. Okay, next, drawing a graph. Okay, so if your observation has data, there's numbers. Okay, there is calculation, right? You're supposed to find the rate of photosynthesis, for example. Then the next question is after getting the result, they ask you to draw a graph, okay, of y against x. So what is y against x, right? You must know how to plot it correctly. Okay, next, state the relationship based on the graph. What is the relationship of your MV and your RV? Okay, so normally, always in an experiment, when you state a relationship, it is a relationship between your MV and your RV. When this changes, how does it affect your RV? When your MV changes, how does it affect your RV? Okay, next. This is the most difficult question. Okay, so next week, I'm going to dedicate one lesson just to give you examples on how to answer questions that need you to define something operationally. For example, define diffusion, rate of diffusion, defining operationally the rate of diffusion. Okay, rate of photosynthesis, define operationally what is the rate of photosynthesis. So this is the most difficult question actually. Okay, but never mind. Next week, I'll dedicate one entire lesson just to give you examples and explain how to answer defining operationally. Okay, next one, predicting. So they will give you a scenario. If they change a certain condition, maybe the temperature, okay, your original experiment, maybe you're doing at room temperature. Then they change the situation. They say they change it to, let's say, 65 degrees Celsius. What is the prediction you're going to get? Okay, I see a question here. 1 minute, yeah. Okay, red block. All right, I always write hypothesis. As MV increases or decreases, the RV increases. Can I say my yes? Correct. This is a very good what we call a format for you to use to write a hypothesis. But not all experiments can be used like this. Okay, later I give you an example lah, right? For example, if I ask you, this experiment is for testing the best pH for the hydrolysis of albumin, let's say, protein. You cannot say as the pH increases, the time taken for the what increases, because there will be, as the time increases, you find that the maximum will occur at 37. After that, the rate will fall. So it's not always possible, right? You will change, you will see according to the question. But I can tell you, red block, everybody out there, most experiments can use this format. Okay, you put your MV there, then you put your RV, slot in there. When this one increases, that one increases or decreases. Okay, in MV increases, RV increase or decrease. Okay, so there's a very good format of the form of hypothesis that you can remember. Okay, so when you see, you apply it to the question. Okay, lastly, classifying. Yeah, classifying is simple. They give you a list of apparatus or materials. They ask you to classify which is apparatus, which is material, and so on. Okay, so I give you each one of these 11 constructs. I will spend some time. Each one, I give you a simple example, and I'll tell you what you need to write. Okay, yeah. So let's get right down to it. So this is an example of a question. All right. So this is I think you may have done it in school, for your maybe your trial exam or maybe your school exams. You are immersing the cut stem of your spinach, all right, into different solution. Okay, so this is a type of question that requires you to write the procedure. Okay, so they don't give you the they don't give you the steps. You have to write what you need to do. All right, okay. And then next question here is state your observation. After you do the experiment, see what happens to your stem, right? Your spinach stem. Then after that, explain your observation. Now, as I mentioned earlier, explain your observation always after observation. It actually means inference. So if the question they change your explain your observation, give an inference. Okay, so a lot of students usually do not understand what is the meaning of inference. So very simple, inference actually means explain your reason for this observation that you get. Why do you get the observation? Okay, inference is the reason for your observation. Okay, right. Now this is drawing your observation. That means your result table. So sometimes your result table can be drawing, or it can be data numbers. Okay, depending on the question. So for this experiment, the what they call the stem, the spinach, that you find that the osmosis occurs and all that. There is no number, there's no data. It is so this experiment this data is called qualitative data, which you cannot measure in terms of numbers. There is no 0.5, 0.6, 0.7, whatever, right? There's no number. So the one that has got number is called quantitative data. So you can either be asked to draw, right? Or you can be either asked to state the color change. That is qualitative. Qualitative means there's no number involved. But quantitative means there is numbers. You need to count the number of bubbles, for example, right? Or you need to measure the time taken and so on. That one with numbers. Okay, yeah. So carry on with the question. You find that every question is only 15 marks. Your experiment, your paper 3 is only 15 marks. Okay, so classify. So classify either your solution X and Y into maybe isotonic or hypertonic or hypotonic. All right, okay. And then fertilizer. This is a scenario. This is prediction. All right, predict what will happen to the vegetables after 2 days if you mix 20 ml of fertilizer solution with 500 ml of water. All right, so prediction, remember they will ask you, you must say what happens first. Then you have to give an explanation why you get this answer. Usually prediction will give you 2 to 3 marks. Okay, so if it's 2 marks, then 1 mark for prediction, 1 mark for explanation. If it is 3 marks, 1 mark for prediction, 2 marks for explanation. And when you explain explanation, you have to base it on theory. Okay, base it on all the theory that you have learned regarding this experiment. Okay, all right. Okay, now we go to each construct. Yeah, right. The first construct here is writing procedure. So what do you need to write in a procedure? Okay, what do you need to write? So your procedure must have only 4 things. Okay, 4 things only. Wajib, because that's why the 4 marks, 4 things here. First of all, you must mention your MV in your procedure, whatever you write. Okay. Then you also need to mention your RV, your responding variable, and also at least 1 constant variable. There can be many. In any experiment, there will be more than 1 constant variable. Okay, but there's only 1 MV. In any experiment, there's only 1 MV. Right, and also precautionary step, at least 1. So here you will have 4 marks already. Okay, let's see an example. All right, now let's say this experiment. You're supposed to investigate the effect of enzyme concentration on the activity of amylase on the starch. Okay, so you are supposed to use the same concentration. All right. Different concentration. enzyme concentration. So you use different enzyme concentration, and then you put it onto the same concentration of this starch, all right? Amylase concentration will be constant, and then you see which one will hydrolyze the fastest. Okay, take the take the shortest time. Okay, what do you need to write? Okay, so let's say this is a question. They already tell you method to handle the variables will be 3 marks: MV, RV, and constant variable. Precautionary step 1. Okay, so here this is what you should, if you are looking at marking a paper, we are looking for these things. First of all, okay, first thing they always ask me, teacher, can I write in active voice or must I write in passive voice? Actually, it does not matter. All right, it does not matter. Of course, if you want to say actually the correct one you should be writing passive, because you are reporting what you have done. Okay, if you write in active, means you're telling people what to do. You're giving instructions. Active is put this, add this, place this, clamp the retort stand or whatever, clamp the test tube, right? Wash the test tube. All this is active. But passive is the apparatus was set up. Okay, you know the BM di oleh di basuh oleh, di cuci oleh, that kind of thing. So all the test tubes were put in water bath at fixed temperature of 37. That is passive. But it does not matter because we are not marking you for your English. We are not marking you for your grammar. As long as the meaning is correct, okay, you would not be penalized. Okay, but make sure the meaning is clear all right? Don't write something that that is ambiguous, that we don't even understand what you're writing. Okay, now MV 1 mark. Now where's your MV? So here you see the different concentration, because you want to see the effect of different concentration. So your MV is, you must do at least a few values. You must do tests for different concentration. Okay, right, then, okay. And also at the bottom here, you will see, repeat steps 3 to 6 for test tube B and C. That is also MV, because you are using it for different concentration, okay, different test tube. So you must show that. If you do that, if you write that, we will give you 1 mark for MV. Okay, red block, here's some question. Pour the concentration into test tube A, B, and C using correct measurement respectively. Yes, but you have to measure what is the measurement. That means the concentration you must mention. Red block. You cannot just say test tube A, B, and C. A, B, and C is not your MV. Labeling A, B, and C, the number of test tube is 3 different test tube. You have A, B, and C, but that is not your MV, because your MV is concentration of the enzyme. So that means you, even though you put A, B, and C, you still have to mention the concentration of the enzyme. So that means 1.0 for A percent, 1.5% for B, 2.0% for C. So you still have to mention. By writing A, B, and C is not going to qualify you to get MV mark. Understand not? Right? You have to state, not just say different concentration. You say different concentration also cannot. You have to mention the concentration that you are given. Okay, right? Okay, so good question. So the more you ask, the more you will learn. Okay, so don't be shy. Okay, RV, what are you going to measure in your experiment? What are you going to record? All right, you are going to record the time taken for the iodine solution to remain yellow. That means once it remains yellow, that means the concentration of the starch has, I mean the what they call the starch has been hydrolyzed. Okay, so red block, yeah, understood, good. All right, okay, the more you ask, the more you learn. Sometimes, you don't ask, you're guessing, right? Actually, you may get the wrong idea. Some people, like you say, you will just A, B, and C, not MV. You must write the concentration, and also number also. Cannot say different concentration. Tak boleh, Okay, what is the CV? CV, you need at least 1, but you can write many, right? Now, concentration, for example, you fix your volume. Okay, you cannot say put some. This is very dangerous. As a science student, you cannot say some. You must specify the MV. The milliliter. How many cm cube? Just give a logical 2 ml, 5 ml also can. All right. And then 37 degrees Celsius. You cannot say just warm water. Warm water is how many degrees? All right. And then you put the concentration. Your starch suspension is fixed. all the 3 samples, like A, B, and C, you use the same concentration of starch. You only change the concentration of the enzyme. You keep the concentration of the substrate same. Yeah, you can only change one factor in any experiment. You can't change everything. MV, you only 1 minute. Okay, right. Next one, precautionary step. 1 minute is the time. Repeat the iodine step iodine test every 1 minute. The diluted water. What is diluted water? Red block. What do you mean by diluted water? Maybe you type in a bit more things I can understand. Okay, precautionary step is here. You have to stir the mixture. Okay, make sure your enzyme and your substrate is properly mixed up when you pour into your test tube. Okay, and then immediately, once you pour inside, you have to take a dropper and immediately extract 1 drop of it to test, see the whether there is starch inside there or not, whether the color of the iodine is dark blue or not. Okay, isolin. Yes, isolin is not diluted water. You call it distilled water. Red block. Isolin is distilled water. Okay, not diluted water. So it's distilled water. So there's no such thing as diluted water because water is just water. You can't dilute it anymore. If it's sugar water, it is manisan. If it's salt solution, then you can say diluted. Okay, because diluted means there is some solvent, solute inside. Okay, right. Let me continue. Right, so that's okay. That's what you can see here. Wait, I'll go back and see. This is what, if you write all that, then you get 4 marks. Okay, you have MV, RV, CV, at least 1 precautionary step, at least 1 CV, you get your maximum marks. Okay, now second constructing a table. So constructing a table, you make sure you must have all your titles. The title is what is the MV you're using. Let's say concentration of enzyme, you must put percent. Okay, or how many mole. Okay, a percentage line usually. Okay, don't forget to write the unit, percent or cm or seconds, right? Do not write your values in fraction. Your table, when you want to do any calculation, your answer cannot 1/5. You must change to 0.2 because later you may need to draw a graph, and then you cannot plot the titik, the points on a graph that has got fraction. It must be 0.2, 0.3, 0.4. Then only you can find the point. You cannot find a point using 1/2, 1/3, 1/4. You can't find it. Okay, so you make sure your data must be in decimal number. Okay, cannot be fraction. Okay, maintain the consistent number of decimal places. Yeah, if your first data is 0.2, no 0 at the back. Then the next one, let's say you got 0.15. Then you make sure your answer on top there must be 0.20. Okay, if your one of your data is 2 decimal places, all the data in the same column must be decimal, 2 decimal places. Let me show example. Calculate carefully, right? Using the given formula. If it's too many decimal places, please round up. Okay, up to maximum 3 decimal places is quite what already. Normally 1 or 2 decimal places is good enough. No need to up to 4. Up to 4, it is quite difficult for you to plot your graph later. You can't find the correct point. Okay, so you must have title, data, and calculation. You may have 3 marks from there. Okay, example, result table, tabulating your data. Right, let's say this is your answer. Okay, so here's your MV. Here's your ORV. ORV is called operating responding variable. Operating responding variable actually means what do you actually record in your experiment. You cannot record the rate of enzyme. That one you calculate. What you actually want to find out is the rate of enzyme reaction. But you cannot see from your experiment, right? What you can actually record is the time taken on it. So your ORV is the time taken for the iodine solution to remain yellow. And then from there, how to find the rate? Rate is always 1 divided by time. Remember, your mathematics also teach you. Whatever you're looking for rate is always 1 divided by time. So here, if it's minute, 1 divided by minute, no? That means 1/18. 1/18, that gives you 0.06. Okay, so you see the consistent number of the decimal places, either 3, everything all 3, or everything in 2. Okay, red block, yeah, typo, so it's the distilled water. All right, so all titles with correct units. All right, make sure all this, you read it across, percentage, minute, and then per minute. correct values. When you do this, your calculation is correct, means it's not uh, outlandish, it's not illogical. You cannot have 1,000 minutes there. All right, your experiment, your 40 minutes also tak jadi 1,000 minutes. So it must be a correct data, and your teacher, your bio teacher will be actually doing the same experiment inside the preparation room to have a set of data to compare your results against. Okay, so she will be using the same apparatus as you are using with the same solutions or whatever. So what she gets, all right, you should be getting at the same time. Okay, if you write something that is really out of the blue, that means something that is really not the same as your teacher's answer, that means something is wrong. Your answer is definitely wrong. They will refer to your teacher's answer first, because she is using the same substance and also the concentration and the alatan that you're doing for yourself. Okay, so they will check it against your teacher's answer. Okay, then correct calculation. Please use your calculator. Right, it's not possible for you to be so accurate when you do your calculation. Of course, it will be wasting time. Please use your calculator, and you must have consistent number of decimal places. Okay, so 3 marks there. Okay, so here, so this is your title, 1 mark here. This is your data, 2nd mark. This is your calculation. If not consistent number of calculation decimal places, consider no mark for calculation. You only get 2 out of 3. So everything must be correct here. Okay, now 3rd construct. Okay, making an observation. Okay, making observation. So how to say observation? You must mention what is your MV here. You also must have your RV in your sentence, and you also must have unit. Okay, example. Okay, give you an example of a question. Okay, here, how do you write an observation? So you don't compare. You don't need to compare. When the enzyme concentration is 1%, the time taken is higher when you use 1.0%. No need to compare. Just make a statement from here. So here you have enzyme concentration as your MV. So give a value. And time taken is also, there is a value here, 80. So make a sentence from there. Observation. All right, answer. When 1.0% enzyme concentration is used, the time taken for the iodine to remain yellow is 80 minutes. Or you can use another one. When the 2.0% enzyme concentration is used, the time taken for iodine to remain yellow is 10 minutes. Any one, all right, any set of data, just write one sentence. Yes. For example, can we write like use different concentration of enzyme? Okay. This one for method to handle. For which one, Gabby? You're talking about procedure, is it? If you say different concentration of enzyme, in the procedure, you must mention what is the different concentration. Okay. If it's way to handle the variable, you will say in your table, here, the table there, you say MV is concentration of enzyme. Method to handle. Method to handle is you say use different concentration of enzyme. Yes, can, correct. Can, way to handle. Or you can, if you don't want to use different, you write back. Use 1.0%, 1.5%, and 2.0% enzyme concentration. Okay, that is way to handle, or we call method to handle. There are two, is it? A table there. If they give you a table, left hand side is what is the MV, you write enzyme concentration. And then on the right hand side, they also method to handle. Method to handle, you say use different can. Or use 1.0, 2.0, 1.5, 2.0 can. Okay, Okay, question. Can we just put the answer without including the calculation in the table? Yes, you can. Answer means like this, is it? You just put the 0.0 something, right? Without 1 divided by 18. Yes, you don't need to show the working. In the calculation, you just write your 0. Blah, 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 blah. No need to show the calculation. Because your answer there is the calculation. It's the marks for there already. You don't need to show your working. Exact amount. Daniel, what does it mean by the exact amount? Maybe you can give me a bit of explanation, then I can understand what you mean lah. Okay, never mind. Let me carry on first. Right. Observation. So let's show you observation. Wait, a bit slow. Next slide. Okay. So like I told you, qualitative and quantitative lah. Quantitative is with value with numbers one. All right, like how many seconds, how many the enzyme, what do you call the rate of reaction, 0.05, got number. Qualitative is without number. For example, this kind of experiment, no value lah. Okay, this one is you need to, like, this is a table with without any value. You show the color change. Okay, you show the color change. You want to see, observe whether in your sample or solution there, in outside the whisking tubing, the water, the solution there, the water, whether then you take out the sample, test it for iodine. If it's still remain yellow, that means no change, that means there is no starch. Then if you take out the sample and then you test it for Benedict's test, you get brick red precipitate, that means there is reducing sugar inside there. Okay, all right. So in the whisking tubing, you have to usually test for inside and outside. This is to show whether your molecule can diffuse across or not. Okay, so if it's big size molecules like sucrose and starch cannot go across. Glucose can go across. Fructose can. All the small, what we call monosaccharide can. Okay. Okay, so this is qualitative data. Color change, you observe, you hear the sound, whatever, all this experiment is qualitative, right? They are picked up the data. Okay, your question? We all only use two kinds of graph for bio. Yes, correct. For bio, normally what we have is a line graph where your data is like continuous, the number, temperature, 10 degrees, 20 degrees, 30 degrees, 40 degrees. It is a continuum, right? You can connect the line, right? That is for line graph. And then for bar chart, bar graph, bar chart. Let's say we are given two solutions to test. You want to test the vitamin C of solution A, solution B, right? Sample fruit A, fruit B. Then you get the answer, you draw the bar chart because they are not connected. Discontinuous data, you learned that in biology in your variation, isn't it? If there's no, this data and this data have no correlation, there's no connection between these two, you can't connect the two lines. You have to put two separate bar charts. Yes, the only reducing sugar is, non-reducing sugar is sucrose. Kitsu. Okay, so reducing sugar, you have 5. Okay, reducing sugar is 5. Non-reducing sugar is 1. Yeah, you wrote from, okay, so non-reducing, correct. Sucrose is the only non-reducing sugar. You have to convert it into reducing sugar, then your Benedict test is positive. Okay, correct. Okay, next one. So this is your MV, this is your RV, color change. All right, now inference. Okay, just now you have your observation already. Now make inference. Okay, for example, inference is explaining the reason why you get this. Okay, I'll give you an example. Right. For example, just now this question, where you get, okay, enzyme, this is your MV, this is your MV, you say you use 1.0%, and then the time taken is 80 minutes, right? Okay, your observation just now here is when 1.0 enzyme concentration is used, the time taken for iodine to remain yellow is 18 minutes. This is your observation punya answer. Now they ask you inference. That means why? Why when you use 1.0% enzyme concentration, why you get the time 80 minutes, which is quite high, which is the longest. Okay, so your inference, your MV and RE for inference is not the same already. Your enzyme concentration just now for observation. Now your 1.0 means is low. Your 1.0 is the lowest, the highest is 2.0. And then your RV, your 18 minutes actually shows what does it represent? When you have a long time for iodine to remain yellow, it means the rate of enzymatic reaction is lower. So for your inference, please don't use the same MV, RV as your observation. You cannot give back the same answer. There's no such thing they ask you the same thing twice, wasting your time, wasting the marks also. So inference cannot use the same MV, that means enzyme concentration. Inference cannot use the same RV, time taken for iodine to remain yellow, wrong. You should be talking about rate of enzyme reaction. Okay, so your inference for this is when the enzyme concentration is low, is it 1.0% is low, ma? The rate of enzyme reaction is low. That means the time taken is long. When time taken is long, that means the rate is low. Understand or not? Right? Don't use the same inference. I always get students answering the same thing again. Inference is not using the same words like you use for observation. Time, okay. Time decimal places. Now, for example, okay, when you use 0.00, you need to see your device. Can your device measure up to 2 decimal places or not? Look at the stopwatch. What is the smallest calibration for your stopwatch? Your smallest calibration, if you are using a digital one, if you're using digital one, yes, you can, because your digital one will have, let's say, 16.32. If you've got 2 decimal places, you're using digital, yes. But if you're using the analog, analog, the old type of stopwatch, when you press one, the press one button here, and then the needle one, got needle, jalan, that one, that one you cannot have 2 decimal places, because the smallest number you have here is 1 second, and in the middle is 0.5. See not? The smallest then calibration, the smallest the smallest line you have is only 1 second. And then in the middle, as the needle is going in between, you get 0.5. So if you're using that analog stopwatch, then your number, you cannot have 2 decimal places for your data. You understand not? Okay. Yes, depending on your stopwatch, some of the stopwatch is 10 seconds, one number like that. You see your own stopwatch, some of the stopwatch may be more more accurate. You have the one line here is 0.2, the next line is 0.4, 0.6. So in that case, you can have your 0.1 if it's in the middle, can. So you check your own data, you check your own instrument, okay? So if it's digital, no problem. If they show you 2 numbers, 0.00, that one you can write down. So you check the stopwatch, see what is the smallest calibration. If the smallest calibration, that one's analog one, you press that one, you cannot have 2 decimal places. Okay, understand? Yes, very good. Okay, so now almost like conclusion. We are not conclusion yet. Inference here is not conclusion. Inference is to explain why when I use 1.0% concentration, why do I get 80 minutes? Because 1.0 is actually low enzyme concentration. Then the 80 minutes means is slow. Your rate of enzyme reaction is slow. So the rate of enzymatic reaction is low. Okay, what is that? Our next student can watch too. Yes, of course, anybody can watch. Yeah, no problem. Yes, already finished also can watch. Of course, maybe most of them won't watch. Because what for? I already finished, right? But anyway, if you watch, good for you to understand this. You may be going to further studies. You go to Form 6, right? You may need this as well. Okay, very good. What's your name? Suzaidi, Ashraf. Okay, right. Next, now listing variables. So can construct of 5. Now listing variable is the easiest one. You can get the answer from your question actually. What is it that you change? Enzyme concentration. 0.5, 1.0, 1.5. They give you all these values, different concentration. That is what is changed. They want you to test this. That is what is different. Ask yourself, what is different? That is MV. What did I change when I do the experiment? Okay, next one. Responding. What do I want to measure? What do I want to observe when I do the experiment? What do I want to test? Test the time taken, for example. Okay, red block. Are there more tests besides finding iodine? There could be, yes. If you are asked to test for a protein, you can use, you may ask a burette test. Okay, we are given the burette test. So you have to go through the 4 types of sugar, all right, starch, protein, and lipid. These are the 4 lah. You won't be asked to test things like nucleic acid, no lah. All right. So these are the 4 things you need to know how to test, lor. All right. You got to go through. That's why you need your theory before you can do experiment. All right. So if you don't know the theory, don't know how to do it, you don't even know. In fact, if you are not given instruction, you wouldn't know what to write also. All right. You need to know how to test. So usually more commonly for enzyme reaction, if they want to test for enzyme reaction, they will either use these 2 things. Starch, they will always use iodine to test. Then for albumin, for protein, protein albumin, they give you albumin, the egg white albumin, they will test it with, normally they won't test it with your protein, the what you call the burette test. They will give you the clarity, see whether the test tube turns clear or not. If the test tube turns clear, that means your albumin has been hydrolyzed. Okay, or they can also twist it, they can also change it. This time they ask you to use burette test. So all your facts must be there. You must know what to do. Okay, next one, constant variable. What is constant variable? What is kept the same in your experiment? You do the experiment a few times. What is it that you don't change? That is a constant variable. Okay, so for food test, only those 4 red block. Yeah. Okay, so for example, here, these are your things, here, these are your MV law. This I show you already. RV, CV, right? All these you must have mentioned in your experiment, on your procedure, right? And then constant variable is definitely more than 1. In any experiment, there'll always be more than 1 variable that you fix. Okay, make sure you fix the volume. If you're only changing the concentration of enzyme, the rest you have to make sure is the same. Otherwise, your experiment is not valid and you can't compare your results. Okay, all this must be CV. Okay, making hypothesis. Okay, just now, who gave me a very good answer? A format, which is your red block, right? Hypothesis must include, in your hypothesis, you must have MV, you must have RV, and you also have the relationship. What is the relationship between your MV and your RV? When this MV change, what happens to the RV? Okay, example, when or as MV increases, I put the color, you can see the color code, right? Blue means MV, red means RV, and then increases, decreases, that is the relationship. When this one increase, what happened to the other one? Okay, either increase or decrease. This is the standard format, but I said, not every experiment can use this format, right? Okay, the higher the MV, the higher or the lower the RV also can. You can also use this format. Okay. First, you must identify what's MV, what's RV. Masuk saja. Just put it inside. You write your sentence. Okay. Then, example. Give you other example of hypothesis. The higher the concentration of glucose, the higher the change in the height of colored liquid in manometer. This one is when the anaerobic fermentation, the anaerobic respiration, right? The yeast, it will produce the carbon dioxide gas. So it will push the liquid in the manometer, the liquid will rise. So that is what you measure. That is your RV. Okay, the higher is your relationship. MV is you change the concentration of glucose. That means first time you do 5%. The second time you put 10%. Okay, that is what you do in experiment. Okay, second example. The higher the temperature, the higher the rate of lipase reaction. Normally for experiment that involves enzyme, there will be an optimum. You cannot say until it means it can go forever. The line doesn't go forever. It goes until 37 usually, around 40 lah. Yeah. So what is this? I did this for last year and yes, ethanol smell. Yes, you will get ethanol smell. All right. The ethanol smell is not the one that goes into your that pushes the manometer. It's actually the carbon dioxide. Okay, it produces carbon dioxide. So that gives the the gas will push the liquid in the manometer and the thing will rise up. All right, next one. The higher the TSA per V ratio, the total surface area per volume ratio, the shorter the time taken for the agar tube to become colorless. I just want to show the relationship between, the rate of diffusion, right? When your agar is smallest agar block, your TSA per V is the highest. compared to a big agar block, okay, a bigger one, the TSA per V is actually smaller if you do the calculation. Okay, so when it's the cube is smallest, you find that the color, right, the acid that goes inside there, you will change the phenolphthalein, it becomes pink color. Sorry, colorless and becomes colorless. That means at first it is pink color, right? So when it goes inside there, it will change to colorless, right? The chemical, the acid, not acid, alkaline. Okay. So this one is haven't come up before. Haven't come up before. So all the experiments that you can expect that haven't come up before can come out, but make sure what you can identify, you can eliminate experiments that takes longer than 40 minutes. Okay. Okay, you have a question there, Ashraf. When you study more, then more knowledge you will get. Yes, correct. Yeah, definitely. It's why teachers always stress you do questions now. Now is the time for you to do questions. Do as many questions, past year questions, niggly papers as much as you can. The more you do, the more confident you will get. Right? So it doesn't matter what question you get, you will know how to tackle it. That's why your time now is best spent by doing questions. Okay, not just by reading, Reading is not so useful now. You got to do questions. When you don't know, go and look up the answer and ask. You must understand. Then do some more questions. Okay, and then you can get the idea already. Okay, now draw the graph. Let's say you already have the data. Now you're asked to draw the graph. Now these are things you need to pay attention. You have to make sure your scale for Y and X is uniform. Uniform means you cannot just jump. Put any number you want. You must be 2, 4, 6, 8, 10. Or 5, 10, 15, 20, 25. You must follow the scale. Okay. Label. You must label both with the unit and the title. Later I show you lah. Mark the origin. Pusat coordinate 0. You must have a 0. Okay. Last time there was 1 year that they penalized. There was child loan. Never write 0 there. You don't get full mark. No. Okay. You must write the 0 there at the base. At the the pusat coordinate lah. That's where you have your starting of the X and the starting of the Y lah. Use a sharp pencil. Right. Draw an X. Not put a titik. Wrong. You don't put a big blob of titik when you draw your wrong concept of drawing graph. Because when you big dot of titik there, big fat one, big bulat, you don't know where's the center. But if you X, you know where's the center. X marks the spot, isn't it? Cikgu Fanida, okay. Your student, Ashraf. Okay, very good. Pandai, Cikgu, budak, your budak semua pandai. Very good attitude also. Okay, do not extrapolate. Apa itu tak? Don't extrapolate. Don't continue the graph. You have your first dot, your last dot here. You don't go and join up suka-suka. I want to extend my line. No, for bio, don't do that. For physics, chemistry, maybe you do lah. You have to ask your teacher lah, because I don't teach physics and chemistry. You have to ask the teacher to confirm whether you can extrapolate or not. But for bio, don't. Okay, and then if your graph is not a straight line, that means you cannot join with one straight line. Please don't use a ruler. You must use a smooth curved line. Okay, and then if you can really join up with one line, please use ruler. Not please, you must. Okay, and read the question whether you are drawing a graph or you're drawing a bar chart. Read the question. Okay. If there's no table, I need something very important. If you skip drawing the table for your experiment, you just straight away, I don't know how you draw your graph there is no mark for your graph because the rationale is you cannot draw a graph without a table. Isn't it? Your table sudah tak ada markah. You think you want to draw a graph, let's say your graph is betul, but still tak ada markah because you tak ada graph, you tak ada table. Okay? If it's a bar graph, you don't have to write 0 as the origin, right? Bar graph, no need. Because bar graph, your X does not start with 0. Your X could be just bread, the calorie, the calorific value is bread. This is for fish, ikan bilis. This one is for kacang. That one no need 0. Okay, but wait. Your Y still got 0, isn't it? Your Y got data, isn't it? Your Y. You better write, hitzu. Because your Y got number. If you got number, you got to start from somewhere. So you better write your 0. Your 0 does not apply to your X, but your 0 will apply to your Y, isn't it? Let's say calorific value. Your X is just the number of sample, all right? Then your Y is your calorific value. That one got 0 one, better right? Huh? Line of best fit, every time must say, okay, line of best fit, please don't use it for bio, red block. Line of best fit is for physics, for chemistry perhaps. You better ask it. Don't use it for bio because we do not skip your point. Okay, I'll show you what I mean. This one, please be careful. You must draw your table. Okay, now look at this. This obviously is not a straight line, correct? Not? You can see all your 3 dots there. All right. So that's why if you see the line of best fit, red block, you will be passing through only maybe 2 out of the 3 dots, correct? Not? You're only passing 2 out of 3, right? That means you already skip some of the points. For bio, don't skip. You have to draw a curved line. Okay. Reaction of enzyme, rate of reaction enzyme is your Y because this is your RV. RV is always on your Y. RV always on your Y. Then your X is your MV. You can also find out which is your Y and your X by reading the question. The first one is always 1 because it's always Y against X. It's always Y against X. It's not X against Y. Okay, this is how they draw graphs. So this is your Y, right? Which is your vertical. Okay, smooth graph. You see the line. This is definitely not a straight line. But please don't use a ruler and don't apply line of best fit. That one is you can use for physics. You ask your physics, just to confirm, but definitely you can't use it for bio. You have to go through every dot. Okay, lengkong mesti licin. If it's straight line, you must use ruler. Not you say you may. If it's straight line, you must use ruler. It means all the 3 dots fall into one line. You must use a ruler. Okay, uniform scale. Make sure you don't skip. You don't skip anything that you want. For example, I don't have a 0.2, so I put 0.3. I don't have a 0.4, so I go and put 0.6. Tak boleh. You have or you don't have the number, you must follow the scale. Okay? Do not extrapolate. Extrapolate means you tambah, you extend your line a bit higher to the up there, and you extend your line bawah, join back to 0. No, don't do that. Okay, so 3 marks here, usually 3 marks. Sometimes they even add things there, right? Your graph also one more thing, please don't only use half the paper. Your graph paper must fill up as big the space on your graph paper. Okay, for example, your graph paper one whole page A4. Your graph is only taking half the page only. No, you must change your scale so that you fit at least 70% of the graph paper. Okay, that also can be penalized because they could add in marks for your graph. There could be 4 marks. Then maybe the extra mark there is for your size of your graph. Okay, right. Now relationship. Okay, we're coming to construct number 8. So how to state relationship? Title, no need title. Usually they don't. Usually some of the paper, the question already give you a title. But if they don't have, you want to be safe, extra safe, you scared you potong marker or you don't get full mark, please write the title. Graph of Y against X, right? What is Y lah? Enzyme concentration, graph of rate of reaction of enzyme against concentration of enzyme, right lah? But usually it's given in the paper, right? If it's not given, please write it. You want to make sure you get your A plus mark, isn't it? Don't leave anything for chance. Okay, state the relationship. How many marks? Sometimes 3 marks, sometimes 3 marks, sometimes 2 marks. Whatever it is, relationship 1 mark. The other one, the next mark is to say the theory. Explain why. Why is the answer like that? Why is the answer like that? Directly proportional to X. Okay, don't do that. Why directly proportional to X? Please use it for your physics. Why directly proportional to X is only you get when it's a perfectly straight line. But most of your graphs in biology experiments, I can tell you, you don't get a perfectly straight line. It's not like your Hooke law, Hukum Hooke. Correct? The extension of the spring is directly proportional to the force, isn't it? That one you can. But please don't use simpan ini for physics. Y is directly proportional to X. Simpan untuk physics, simpan untuk chemistry. Don't use it for bio because it is not, you won't get a straight answer. Most of it. Okay? Simpan untuk physics saja. Okay, next. So, relationship. Okay, example. Now, when you increase the concentration of enzyme, why the reaction is higher? Because, okay, sample answer. The higher the enzyme concentration, the higher the rate of reaction. This is your relationship. I can get from the graph. You see the graph going higher and higher, the line is going higher and higher and higher. That means the higher the enzyme concentration, the higher the rate of enzymatic reaction. 1 mark. Then explain why. Explain why is the E1, E2 lor. Because when you increase the concentration, there are more enzyme molecules to collide with the substrate. Okay? More enzyme molecules will collide with substrate. There will be more, they can bind to the substrate. Therefore, more enzyme substrate complex can be produced or is formed. Okay? Understand not? So you have one, you must state the relationship first, 1 mark. Then you say why. Because you got more enzyme, you can collide more. Then when you collide more, you can form more the enzyme substrate complex. So you get more hasil, faster, right? Okay, operationally, this one next week I'm going to devote some examples of defining operationally, but 3 important ideas when you want to answer this question. What is X? They always ask you, what is rate? Define rate of diffusion operationally. What is X? What is rate of reaction? Rate of diffusion based on this experiment. Understand this, based on this experiment. That means your answer must be only for this experiment. Cannot write an answer that is general. Cannot. Your defining operationally answer is not a general answer. It's only boleh pakai for this experiment sahaja. Okay? How is X measured in this experiment? What is your RV? You also mentioned your RV there. Okay, how do you measure your X? All right, what do you see? What do you measure? And what affects your X? That means what affects your rate of diffusion? What affects your enzymatic reaction? That means you need to mention RV, MD lah. Okay, example So this one, don't worry. Next week I'm going to go through again. So defining operationally, examples, some examples, a few examples. Rate of diffusion is the process of movement of hydrochloric acid into agar tube per time that is determined or shown by the time taken for agar tube to become colorless. You see, the first one is what do you use in the experiment? You actually let your hydrochloric acid move into your agar and you measure the time. That is the rate given by this one. And what is RV? How determined by what? Determined by the time taken. This is what you measure. Time taken for the agar to become colorless. Okay, so when the hydrochloric acid went into your phenolphthalein, it will become colorless because phenolphthalein is pink color, right? Phenolphthalein, when it mixed with the alkaline, it becomes pink color. So when your hydrochloric acid diffuses inside, it will turn colorless. That is experiment. Okay, rate of diffusion is affected. This is your MD. Is affected by the ratio of TSA per V. Okay, later, next week, I'm going to give you examples, show you the experiment. That only you can understand better. All right. Transpiration. What is transpiration? Define transpiration operationally. Transpiration is the loss of water from balsam plant. You see, not just general plant. You see, you must mention in the experiment, kalau you pakai balsam plant, you must say balsam plant. If you say hydrilla, if you use hydrilla, you must mention the name of the plant. That is why defining operationally, people don't really get the concept because tak ada satu jawapan yang sama for all the experiments. Understand or not? X is experimental theory that is influenced by MV. Okay, you forgot the full sentence. Roughly like this, roughly like this. Okay, red block. Yes. Yeah, like that, correct. Okay. Now, next one, determined by the change in mass of potentiometer in 30 minutes. Your experiment, you are supposed to measure the change in the mass of potentiometer in 30 minutes. Okay, 30 minutes, you see how much water already evaporated. That is your second point there. Then affected by what? Affected by air humidity. So in your experiment, maybe they change the air humidity lah. So depending, it is very, very, very dependent on your specific experiment. If you want to get your full mark, there's nothing theory. You cannot give a definition secara theory. Defining operationally must be based on that particular experiment. Understand? All right. Okay, I have another question here. Bell time. Can I change a bit the theory mentioned in the instruction or question for the operation? Because sometimes the question will give the theory. If the theory applies, whatever you write for your operational definition, Bella, you have to make sure you got mention your MV, your RV in your experiment. So you see all this is very specific, isn't it? Not every experiment using the 30 minutes, not all using Paulson plant, not all experiments are using the hydrochloric acid. You see, you must, what chemical you mention, you must write also now. So your theory, if it's given there, you don't write the theory already. Your question given the theory is definitely not defining operationally, because defining operationally is not theory. Okay, it's not theory. So I think it's very specific. Okay, next week I give you examples of questions. Maybe you can understand a bit better. See the experiment set up, then I give you the answer for defining operationally. Maybe you boleh catch it. Yeah. Diffusion. Okay, what is the operational definition for diffusion? Diffusion is, always answer is. Something is. Something is. Your sentence must start with that thing they ask you is. Because when you want to define something, people ask you what is a mask? Then you say mask is. You have to say is. What is paper? Paper is, isn't it? You have to answer with the word is. If your question doesn't start like that, salah already, because you are not defining. Okay, defining. Okay, diffusion is the process of water molecules moving out of the whisking tubing. You see, in the experiment, ada pakai whisking tubing into the sucrose solution. You also pakai the sucrose solution in the beaker, right? It is determined by the distance of X to Y. So inside the experiment got the tube there, the capillary tube there. You measure X to Y. That's why everything must masuk inside your defining operationally in order to get full mark. Whatever is inside the experiment, very specific answers must go inside here. Okay, and then diffusion is determined by what? The concentration of sucrose. That means in experiment you change the concentration. Okay, we do one time maybe 1%, then do a second time 2%, 3%, 4%. Then you find the distance is different. Okay, okay, three more examples. Okay, this is BM, sorry. BM only. BM, those who want to see, you can slow down your video later, then you can see lah. Okay, I will explain in English because, taking a bit of time. Okay, prediction. Okay, second last construct. Prediction. Prediction is quite easy lah if you know the theory lah. Let's say if you're talking about enzymes, you must know that when you increase the temperature, all right, your enzymes will stop working. Isn't it? Temperature is too high, for example, up to 50 degrees Celsius. You must know that the rate of reaction will be slow already, okay, because of the theory. So predicting, you can fall back on theory, yes. Okay, so 1 mark for prediction with the correct unit, and the other mark, either the other 2 marks or 1 mark, you must explain the theory. Explain the prediction, but this is based on theory. Definitely, because this is theory. This is nothing to do with it's always fall back on the theory. Okay, prediction must be stated. If you don't have prediction, you straight away give explanation, no marks. Because it's not logic. People ask you something, you never answer. You go and answer something else, isn't it? You must answer that question. You must state your prediction first. Okay, example. 1 mark, correct prediction. If you have 3 marks, if this question is 3 marks, then you will have 2 marks for your explanation. Okay, example. For example, if you increase the temperature for your this orange juice, okay, when you want to test your DCPIP, you go and boil your orange juice, or you slice open your orange and you let it oxidize in the udara, right? That means the oxygen will decrease. So the volume of the juice needed to decolorize DCPIP will increase. Okay, becomes higher than the original value. Whatever original value that you are asked to compare, you have to say higher than what. Okay, because explanation, heat destroys, heat denatures vitamin C. That is theory. If you don't have theory, you cannot answer this. You can only give a blind prediction only. Okay. Next one. Why, or another answer is the concentration of vitamin C in the juice decreases. Okay. So you explain why you will need more volume of juice to decolorize the DCP acid, because it's no longer so much vitamin C inside there already. Okay. Or you can say the concentration of vitamin C in the juice becomes lower than something, all right, because they never ask you predict what. Either you predict the volume of juice, or either you predict the concentration of vitamin C also can. Okay, see the question. They just ask you predict only, predict the result. They never ask you predict the volume of juice. If they ask you predict the volume of juice, you must say the volume of juice. Don't go and say the concentration of vitamin C lah. Right? See the question. Always underline what you're supposed to answer first. Ah. Oh, what is DCPIP? They're the dichloro phenol indophenol dichloro, the substance that you're supposed to put your vitamin C inside to test your juice got how much vitamin C. Go and check back. This is Form 4 work, chapter 6 for nutrition. Form 4, Form 4, chapter 6. DCPIP is a substance that actually you add in. If you want to test how much vitamin C your juice has, you must use DCPIP. Okay, dichlorophenol indophenol. Okay, check back. Then maybe ask your teacher to explain back this one, or you check back first. This one you will have learned already. Okay, because again the same reason, always talk about denaturation when the what you call increases, the what increases, the heat, the temperature increases, it will actually denature the vitamin C, destroys or denature vitamin C. Therefore, the volume of juice needed to decolorize increases, so you need more juice to decolorize your DCPIP. Okay, now, so this is your prediction, the prediction question. Let's say I immerse the test tube N. This is the same experiment just now, the concentration of enzyme. I put 65 degrees Celsius. So 3 marks, let's say. So the time taken for iodine solution to remain yellow will be longer than 30 minutes. See, longer than the original. Let's say the original was 30 minutes. Now, if I change the temperature to 65, definitely be longer, because you already destroyed the enzyme. The enzyme already denatured, so it doesn't work anymore. Okay, so this is your must have value. Cannot just say increase. Cannot just say higher full stop. Higher than what? Okay, higher than what? Okay, explanation. In high temperature, the rate of enzyme activity decreases or stops at 65 degrees Celsius. Enzymes are denatured. The active sites for the reaction has changed. So that is your explanation based on theory. Okay, right? Okay, last is the easiest question. Easiest question is to classify. Okay, let's say they give you a scenario based on your diagram they're given in your experiment. Classify all the apparatus and material given to you to two groups, whether it's apparatus or material. Okay, remember how to identify what's apparatus? Apparatus is something that you can reuse, something that you don't pakai satu kali and then you throw away. It's too wasteful, like beaker, burette, all these you don't throw away. You can wash, you can reuse. Okay. Material is something that is usually once you use it for this experiment, you can't use it anymore because it has altered, it has changed, it already becomes something else, or maybe something that is so cheap that you don't need to reuse it. For example, whisking tubing. Whisking tubing, can be a material because once you pakai your whisking tubing, we are not going to wash it and use it again, right? You can throw it away actually, right? So depends on how you use it once or you can use it again for another experiment. Okay, whether you want to classify, maybe they give you a few things here, either high rate or low rate. Okay, they give you scenario lah. Okay, certain cases you classify this one high or low, right? Then maybe food class, they give you a few example of food. Which one is classified under carbohydrate? Which one is lipid? Which one is protein? Okay, and then MV or RV, they ask you which one is MV, which one is RV. All right. or type of solution like isotonic, hypertonic, hypotonic. So this is one of the easiest questions actually. Okay. So so now, okay, it's the most interesting part of the lesson. What kind of questions can come up? What kind of experiments can come? Everybody asking me. Okay, very simple. Only experiments that can be conducted within 40 minutes. You can go and check through all the experiment in your textbook from 4, from 5. All right. Things that experiments that take too long. Okay. That is not possible. That need you to go into the padang. Need you to go into the toilet to take your urine sample. Not possible. Tak boleh buat kan. Okay. So experiment, everything can be completed in 40 minutes. Okay, so these are the possible experiments. But this number one, I would say unlikely because most of the schools don't have good microscopes. The microscope need to be serviced every year, and sometimes lacking of good microscope, you can't really see very well. But you can prepare. Maybe ask you to prepare. But prepare not seeing also like quite useless as well. Okay, next question. This is simple experiment. Studying the movement of substances across a semi-permeable membrane, like vacuum tubing, osmometer, and so on. This one also can be done within 40 minutes. Okay. Studying the effect of different concentration of solution on plant cell, like immersing your potato strip, immersing your stem, your cucumber strip, whatever, into the different types of solution. Iso, hypo, hyper, like that. Okay, that was simple. I think you've done it for your uh, exam in school. Okay, studying the effect temperature, this one, temperature or pH on the activity of enzyme, amylase. They will always use they always use albumin for amylase, and then pepsin for pepsin is enzyme. All right, amylase you will use starch, pepsin you will use albumin. Okay, next one. Okay, next one. Next list. Okay, investigating the respiration or alcoholic fermentation on this in yeast. Okay, now this one came out before actually. Okay, I'll tell you which one came out before. Studying the digestion of starch, protein, and lipid in food samples. This one using Benedict solution, using the starch and so on. Right? And studying energy value. Burn the food sample, burn your bread, burn the ikan bilis, burn the kacang. Calculate how much energy is released by the burning food. Okay? Then this one, Hitsu. This one they're using determining the concentration of vitamin C in your juice that is given to you. This one needs the DCPIP. This one, since you're not sure about this one, means that this is something that you need to read. If it doesn't come out, Ujian Amali Sains, it can come out in your paper 2, so you need to know what's DCPIP. Just read up. Okay, estimating, this one never came up before. Estimating the size of the organism using quadrat sampling. This one not really go to the field and put the quadrat. They give you a piece of map, right? And then I think I did video before last 2 years or something. Please go and check my video in my channel. I've done video on quadrat sampling before. Okay, how to do if the question come up, quadrat sampling or CMRR. Okay, capture, mark, recapture, release, and recapture. Okay, use beans. Simulation means you're not going to capture, you're not going to go to the field and capture ants or seafood babi. No, we're going to give you a pail, put sand, give the biji kacang representing animals. You go and scoop up. This is interesting. Okay, next one. Studying variation, continuous and discontinuous variation in humans. Simulation also, they give you a picture, different types of earlobes, different types of fingerprint also can. Okay, and 11. This one came out last year, so I put in there. Studying the rate of photosynthesis using an aquatic plant, usually hydrilla, right? And then you measure the bubbles that is released. Okay, let me tell you what has come out before. This one came out 2022, right? This one came out 2023. This one came out 2021, right? This one came out 2024. All right. So now, already come out, I would say highly unlikely to come out again. I'm not saying that it won't come out, but don't take my word for it. Doesn't mean that you don't have to study. You need to know in case it comes out. But what I feel, there's so many experiments that haven't come out before, quite unlikely lah. Right? So up to you to believe me, or up to you not to believe me. Just read lah. Okay? What I've given to you, I've shared to you today is all the steps how to answer when you have this construct come out. all the 11 types of questions. All right, so I will see you next week, hopefully, where I have specific examples of defining operationally, where you will see how to get your answer based on the experiment. Okay, I hope you can join me next week again. All right, so this is my channel. All right, if you want to see, I've done quite a lot of videos. If you want any help on any particular topic, please go and search through it. I've also done some in DM when under Academy YouTuber. Okay, so you check through out what you want. All right, this is my channel, www.youtube.com, the Chong. And if you need to contact me, you want to ask questions, I'm most welcome for you to contact me. All right. So this is my Telegram, for that. T.me Telegram. So you can contact me if you want to ask me any questions. No problem. All right. Okay. Farida, you're still here. All right. Okay. Thank you very much for joining me. All right. So thank you. Yeah. So please ask the teacher was DCPIP. Yeah. Please Cheng Mi Man Ho Gam Hong. Okay. Everyone. Okay. Thank you, everyone. And I hope to see you next week where I'm going to explain defining operationally in detail. Okay, so please check out my channel. If you have anything else that you need to see, please go and go through lah. And you can also share the links to your friends. Okay, bye bye everyone. See you next week. Okay, wait, let me find the the music. No music. Okay, where is it? Okay, right. Bye bye. See you. Where is it? Okay, right.